So, this is Patrick. And like most respectable plants, Patrick has the unique ability to convert carbon dioxide into useful oxygen, which is important for, for us to breathe and consume, which allows us to live. So it's pretty important that Patrick can do this for us. And not only can he do that, but he also is an autotroph, which basically means he doesn't need to go out to McDonald's for every meal, unlike we do. But how does this actually work? How can Patrick make food for himself? How can Patrick convert the carbon dioxide into oxygen that we can then breathe? Well, in order for that to happen, we're going to need to take a closer look at his leaves. This is a plant cell, and if we look at the plant cell, we can see different chloroplasts within the membrane of the cell. Within these membranes, the light-dependent process is going to take place. This is the process in which producers like Patrick convert light energy into chemical energy, like glucose. Let's zoom in even further to get a look at these chloroplasts. So this is a chloroplast zoomed in. So within the chloroplast exists stacks known as grana. Each of these stacks is known as a granum, and each granum is made up of multiple discs known as thylakoid, as we can see here. Within each thylakoid membrane, there are specialized membranes. Within the thylakoid, membranes have specialized pigments called chlorophyll, which is responsible for absorbing photons or light energy, which will kick off our process. At the end of this process, we will be left with adenosine triphosphate, which is important, even necessary, in all living things, as it provides energy for all chemical functions in plants and animals. Not only will we end up with ATP, but we will also end up with the coenzyme NADPH, which will serve as an electron carrier for the next process. So let's zoom in into one of these thylakoid to take a look at how we end up with these useful products. This is the thylakoid membrane, where the process will take place. Let's label some key points that we can keep track of as we move through the process. So if this is the membrane, this side of the membrane is known as the stroma. And within the membrane is a gel-like substance known as lumen. It's also referred to as the thylakoid space. Another important structure to keep track of is this structure here, which is known as the photosystem 2, shortened for PS2. And no, I'm not talking about this PS2. As well as the PS1. Again, not the video game. What both of these photosystems is that's where the concentrated pigment of chlorophyll is that will receive the photons from the light that will start the process. Now Patrick is getting hungry, so let's get him some glucose, which requires the formation of ATP and NADPH in this process, carried on to the next process. So, to kick off this process, we need light from Cool Mr. Sunman. And photons from the light source will reach the thylakoid membrane into the PS2 here. The photons will reach the chlorophyll concentrated area of the PS2, which is then absorbed and passed along to a reaction center. And in this reaction center, we have multiple electrons, and after they receive these photons, they become excited. These electrons get so excited that they leave the PS2 and reach an electron acceptor. The two electrons that existed in the PS2 are replaced with other electrons that are created through a process known as photolysis, which includes the breaking down of water molecules, but we will cover that in the next video. From the electron acceptor, the high-energy electrons will transfer across a series of electron-carrying molecules. These molecules altogether are known as the electron transport system, or the electron transport chain, ETC for short. As the electrons move along the electron transport chain, the electrons create small amounts of energy. The energy release causes hydrogen ions that exist in the stroma of the thylakoid to be pushed through this structure, known as the hydrogen pump. Hydrogen pump. And into the lumen of the cell, where they become concentrated and will not be able to pass through the way they came because this membrane is impermeable to these hydrogen ions. While this process is taking place, the photons from Cool Mr. Sunman reaches the PS1, where other electrons become excited and start moving along another electron transport chain. Whereas the electrons that existed in the PS2 were replaced through a process known as photolysis, the electrons in PS1 become replaced by the electrons that traveled from the PS2. So these ones will travel and take the place in the PS1. The electrons will then be accepted by another electron acceptor and produce a substance known as NADP+. If we have NADP+ that exists up here, these electrons are going to reduce it. To reduce NADP+ means that they will each gain an electron. Once they have each gained an electron or become reduced, they will have become NADPH, which is one of the main products that we were seeking for. These coenzymes will carry on into the next cycle, the light-independent cycle, so we will see them again shortly. Now we can't ignore our hydrogen ions. These hydrogen ions need to move across their gradient, but simply can't because this thylakoid membrane is impermeable to these highly active ions. A special structure known as the Atp Synthase, which is located right here. Let me just adjust it there. The Atp Synthase, it's an enzyme. This structure, which is embedded in the thylakoid membrane, is the only way that the hydrogen ions can get across and move across their gradient. As these ions diffuse, and as the energy created by the gradient helps to create a bond between existing phosphates and ADP, or adenosine diphosphate. This form, when these two create a link, we get ATP, which is the other major product we were looking for. Now, the involvement of hydrogen in linking the phosphates to the adenosine diphosphate to create adenosine triphosphate is known as chemiosmosis. So we have our adenosine triphosphate and we have our NADPH. Now Patrick is a little bit happier, and that is the light-dependent reaction in a nutshell.